Okay, this lecture is to help you understand when I talk about gene expression, what I'm really talking about. And basically it's making proteins. So in eukaryotic cells, you have two steps to gene expression. You have transcription, which is the copying of DNA to RNA, and that involves the enzyme RNA polymerase. You'll see RNA pol 2. So eukaryotic cells have 3 different RNA polymerases, and our mRNA is transcribed using RNA polymerase 2. Translation, ribosomes. So for all viruses, translation happens the same. It uses the host cell ribosomes. So what we're going to talk about in this lecture is, one, if you're a DNA virus, how do you make your mRNA? And remember, on a long chromosome of DNA, you'll have thousands of genes, and there's a promoter. Oh, I hate this. There's a promoter that, along with transcription factors, recruits the RNA polymerase. And then the RNA polymerase copies one strand of the DNA. RNA polymerase makes sure there is a 5' cap and a 3' polyA tail on all mRNA. And then also in eukaryotic cells, introns are spliced out, exons are spliced together, and sometimes RNAs are alternatively spliced, which means perhaps one of the exons is not spliced in, and that gives you different proteins from the same mRNA. RNA polymerase and transcription happens in the nucleus. So for DNA viruses, it's pretty straightforward. We're going to assume the double-stranded DNA is made. We've talked about that in a different lecture. Gene expression, the transcription, I should say, happens in the nucleus, just like with eukaryotic cells. I'll show you poxviruses, they're an exception. The DNA viruses are going to use the host RNA pol 2, so that means they're already going to get a cap and a tail put on so that the host ribosome can recognize them just fine. They may use splicing. The virus needs to supply a transcription activator. So these transcription factors, the virus needs to supply proteins that help the RNA polymerase recognize the viral DNA for transcription. The other thing that viruses do is they control when genes are expressed. And this is especially important in DNA viruses. So, for an example, human papillomavirus has early and late gene expression. So it controls when it's going to produce the capsid. And that's later because that's needed later in the assembly process than when it's going to do replication of its DNA. And so I want you to see here that human papillomavirus encodes some of its own transcription factors. So this is how it's regulating, this is how it's telling the cell, hey, come and transcribe my gene to make mRNA that can go on and get translated. I throw up SV40 polyomavirus just to show you that yes, some viruses use alternative splicing. Okay, so the benefit of alternative splicing is that you can have a smaller genome but still make a more or a variety of proteins by using different open reading frames and using different exons to put together proteins. So, in general, DNA virus is really straightforward. They use the host RNA polymerase. It puts the cap and tail on so that the ribosomes translate it just like they would a eukaryotic mRNA. Nothing that exciting. Poxviruses, as I said, are the exception. They have a large genome. They encode about 200 proteins, or over 200 proteins, I should say. And they do everything in the cytoplasm. Because they encode their own RNA polymerase. But they still use the host ribosomes. So, same terminology though. They have a DNA genome. Transcription is to make the mRNA. Okay. Translation is making the protein. So you've got transcription, and they also have early, intermediate, and late. So they have actually 3 timings for gene expression. Retroviruses, we talked about that they actually integrate their genome into the host chromosome as double-stranded DNA. So retroviruses produce their mRNAs using the host RNA polymerase. So in this sense, even though they start with an RNA genome, they express genes just like a DNA virus. Okay, so I've mentioned the cap, I've mentioned the tail. The 5' cap and the 3' polyA tail are essential for translation. They're important for some other things too, but we're going to focus on translation. So that means they're essential for the ribosome to recognize the mRNA. Okay. RNA viruses do not use a host RNA pol2 to make their messenger RNAs. They use RNA-dependent RNA polymerases, which means they're not automatically going to get a 5' cap and a 3' polyA tail. They're still going to use the host ribosome. So somehow they have to have the host ribosome recognize their messenger RNA. And so that's what we're going to talk about, a few strategies for RNA viruses. So, for example, Rotavirus, which is a double-stranded RNA virus, it makes its mRNA, and it actually puts on a cap, but it has no tail. And so the way Rotavirus deals with this is it produces its own protein called NSP3 that protects and binds the 3' end of its messenger RNA and recruits the rest of the cap binding complex. So if you look right here, here's part of the cap binding complex. And so even though Rotavirus does not have a tail, it can still use the host ribosomes for translation by using its own protein to make this ribosome recruitment complex. So here's the cellular, and here's the rotavirus one. Okay, so a unique strategy for this type of virus. Some viruses have a different strategy. They use what we call an internal ribosome entry site, or IRES. And this is like a landing pad for the ribosome and translation factors. Okay, so poliovirus does have a small polyA tail, and it has a cap, but it's a protein cap. But that's not necessary for translation. This is more for stability of the viral RNA. So what this structure does is recruit the eukaryotic translation initiation factors, and then it recruits the ribosome, and the ribosome binds right onto the AUG and starts translation. Now, poliovirus is a positive sense RNA virus. So in fact, the RNA genome that is packaged in poliovirus can immediately be translated. Rotavirus is a double-stranded RNA virus, and inside its capsid, the RDRP makes mRNA from the double-stranded RNA genome. So again, RDRP is making this. That's why it doesn't have the tail, in this case, like a normal messenger RNA. Poliovirus, the positive RNA we talked about in the previous lecture, is made by a different polioviral RDRP. And that's why, again, it doesn't have that same 5' cap, but it overcomes the need for a cap and tail by using the internal ribosome entry site. Poliovirus even shuts down what's called cap-dependent translation. So poliovirus does cap-independent translation, and it shuts down the host translation by cleaving one of the cap binding protein complexes. And so the host mRNA can no longer be translated, and all the resources go towards poliovirus. And this is pretty common with RNA viruses. So this is showing you in the red circles are different viral proteins, and they're showing you what they target to shut off host translation. So there's this competition for DNA viruses and for RNA viruses to have the host become a viral producing factory. And so viruses use different strategies to recruit the host machinery and recruit it preferentially over the host producing more of its own proteins. Poliovirus is also made as a polycistronic, or is called polycistronic gene expression. It makes a poly protein, and remember, poly means many. So in fact, it's translated as one long protein. And subsequently, that protein is then cleaved by viral proteases into all these individual proteins. This means that poliovirus proteins are made at a 1-to-1 ratio, right? So there's one of these, and one of these, and one of these, and one of these. And we'll see not all viruses do that. Okay. Some viruses do something called making subgenomic mRNAs. Right? So if this is the genome, then they transcribe multiple different sizes of RNAs from one genome, and then these go on and get translated to individual proteins. Okay. So again, another way to regulate gene expression, which is your ultimate goal, making proteins. A lot of these strategies I want you to see is that many RNA viruses have very small genomes, but they can put a lot of information into those small genomes by using things like subgenomic mRNA or something called ribosomal frame shifting, where you're changing the reading frame by having the ribosome either move back one or move forward one. And this makes what we call a fusion protein, like for HIV gag-pol. And then that fusion protein, just like with the polio proteins, can get later cleaved into individual proteins. But again, it's a way to keep a compact genome being able to express multiple proteins. So instead of having to have a new ribosome entry site or a new transcription start site, it can just do a frame shifting during translation and get two proteins produced. Some viruses have multiple RNA genomes, right? And that's fine because each genome can be transcribed and translated separately. Where this becomes an issue or a challenge is in the packaging to make sure you get all of these different genomes. But the genome replication is the same whether it has one RNA segment or multiple. I should say the genome replication and the gene expression, because we're talking about gene expression here. Influenza is an example of a virus that actually steals caps. So to deal with that issue of having a 5' cap, the virus's RNA-dependent RNA polymerase actually grabs host mRNA, cell mRNA right here, takes that cap and uses that as the start for synthesis of viral messenger RNA. So kind of a cool strategy to avoid that need for its own capping enzyme. But the viral mRNA has the cap, so it's going to be translated just like the host mRNA. Rhabdovirus has a single genome, but unlike poliovirus, it doesn't translate one long polyprotein. Instead, it does something called sequential transcription, which means you're going to make multiple mRNAs, which will then be translated. The key here is that the RDRP, which is going to make these mRNAs, can only start at the very 3' end. And so it has this series of start, stop processes. So it will start and it will make the mRNA for protein N. It will stop and it may fall off or it may continue going. And so between each gene is a stop, restart sequence. And very rarely does the RNA polymerase, RDRP, make this mRNA and make this mRNA and make this mRNA and make this mRNA and make the lmRNA. So the sequential transcription means that the genes at the very 3' at the beginning of the RNA are going to be made in higher levels than the genes at the end. And these genes, the N is for the nucleocapsid. So it makes sense that you need lots of this protein produced and less of L, which is the RDRP produced. So it's a really cool strategy to save resources. Unlike polio that's making everything at a 1-to-1-to-1 ratio, Rhabdovirus with sequential transcription can make more of one gene and less of another. So what's the take-home from all of this? The take-home is that gene expression involves transcription which is the production of mRNA and it involves translation, which is the mRNA sequence being read by the ribosome and protein being made. Okay. This is pretty much the same for any mRNA. The difference is how you recruit the ribosome. We talked about some of those strategies if you don't have a 5' cap and a 3' polyA tail. And then transcription is really where you get control of gene expression. So how are you producing those mRNAs? Are you using the host RNA polymerase, like DNA viruses? Are you producing subgenomic mRNAs? Are you doing sequential transcription? Are you just transcribing the whole polycistronic gene sequence? So lots of different ways to control the levels of viral proteins made. And again, it's this competition of resources between the host cell and the viral kind of production. Yeah. So hopefully that helped. Thanks.